Good morning, everyone. My name is Brian Duke. Thank you for attending the last presentation this year of the California Oil Spill Study and Evaluation Program, also known as COSSEP. This presentation is being recorded and will be available on the COSSEP webpage in about 4 to 8 weeks. Today we have a presentation by Ellen Faurot-Daniels and Bryn Phillips. Ellen will provide more information regarding Bryn's background and her background. Speakers request that all questions are held until the end of the presentation. At the conclusion of the presentation, I will moderate a question and answer session. Please submit your questions using the chat option of the Microsoft Teams, and I will read the questions to the presenters. Also, please note that some advertisements mistakenly reference this presentation as an injury assessment of shoreline. This is not correct. Thank you in advance for your cooperation. Without further ado, I present Ellen Faurot-Daniels.
Well, hi everyone, and thanks for joining us today. As Brian said, I'm Ellen Faurot-Daniels. I'm a Senior Environmental Scientist for OSPR and California's state lead for Applied Response Technologies, which we abbreviate ART. ARTs include the use of biological or chemical treatments, broadly called oil spill cleanup agents, and they're abbreviated OSCAs. In addition to leading OSPR's work on the policies that underlie use of ARTs, I do the state's OSCA product licensing. I'll be joined today by my co-presenter, Bryn Phillips, who leads the UC Davis Marine Pollution Studies Laboratory at Granite Canyon. That's not at Davis. It's located on the coast about 9 miles south of Carmel, California. Bryn's lab did the toxicology assessments, and he'll address the test methods and resulting data. The OSPR Petroleum Chemistry Lab in Rancho Cordova was also involved in this project. They did the total petroleum hydrocarbon analyses in water samples sent to them by the Granite Canyon Lab. So Bryn will also cover those results. The pictures on this first slide will give you a preview of what will follow. Chemical dispersants are just one type of OSCA. Corexit EC 9500A, also known as Corexit 9500 or 9500A, is the dispersant most commonly available for treatment of oil spills in U.S. waters. Oil spilled at the surface will get physically mixed by waves and chop into the water, even if we don't add dispersants. Dispersants can, however, kind of jumpstart the process by breaking oil into much smaller droplets that are then more readily mixed into the large water column beneath the slick. The smaller droplets are less likely to stick to each other and float back up to the surface or stick to other surfaces. They're in smaller, bite-sized pieces for the oil-degrading microorganisms that actually feed on them. Shifting some of the oil from the surface and into the water column could result in significantly less oiling of marine birds, turtles, and mammals that live, feed, and travel near the water surface. These are also some of the same species with long lifespans and low reproduction rates, so it's harder for the ecosystem to replace them if they die in a spill. But other organisms and life stages that are in the upper water column, and we're talking here about water down to about 10 meters, 30 feet, and especially the upper 1 meter, they'll be exposed to the toxic oil compounds in that plume of dispersing oil. And if that exposure is at a high enough concentration or for a long enough time, they will suffer the negative effects. So our challenge is to understand what's likely to get exposed, the types and thresholds for these exposures, and to balance the pros and cons of dispersant use in any spill response. Although we would not recommend using dispersants for most of the oil spills that may occur off our shores, there are a limited number of cases in which we would. So first, welcome to my weed patch. You'll see just some of my weeds from home and hear about just one weed patch from work. You'll need to follow me into that work weed patch at least a little bit to know why we did this COSSEP project. Before the use of any OSCA product, whether a dispersant, washing agent, a sorbent, an oil solidifier, a bioremediant, etc., that product is reviewed by both the federal EPA, and in California by OSPR. The federal EPA spells out their testing requirements under Subpart J of the National Contingency Plan. Those federal testing requirements vary depending on the OSCA product. The EPA does not license an OSCA, but it will list it on the National Contingency Plan's product schedule once it verifies that the information provided by the manufacturer is correct. That product isn't available for a federal on-scene coordinator, such as the U.S. Coast Guard, to consider for an oil spill response. There are other levels of approvals needed before the FOSC can actually use the product. Product runs the federal gauntlet to get on the product schedule. It needs to go through California's state licensing process before it can be used on an oil spill that is in, near, or threatening state surface waters, whether marine or inland. In California, dispersants are currently only considered for use over deeper marine waters. OSCA, can't click properly here. OSCA testing procedures, particularly those for dispersants, are slated for update at the federal level, and state licensing requirements may follow suit. And I'll just say that someone is not on mute. Let me tell you a little more about the California program for the review and licensing of OSCAs and about dispersants in particular. California is the only state with a separate OSCA licensing program, but it didn't originate with OSPR. The State Water Resources Control Board had been licensing OSCAs for several decades before OSPR assumed the OSCA licensing program from them in 1996. OSPR's new regulations and licensing guidance documents created in 1996 kept all the good stuff inherited from the State Water Board and then established parallel or comparable processes to those set by the federal EPA. At that time, California went above the federal standards by adding a modified test for dispersant efficacy, adding toxicity testing on red abalone larvae, and using discharge thresholds for trace metals and total chlorinated hydrocarbons as established by the State Water Board. Our practice has also been to consider our Fish and Game Code when looking at a product's potential to impact the environment in which it's used. Even products considered non-toxic can create other hazards to watch out for, such as using an otherwise non-toxic product in a way that poses an attractant and ingestion hazard for wildlife. Dispersants are the OSCA that get the most attention. Unlike other OSCAs, we can't recover them from the environment once they do their job. We can't really see that plume of small oil droplets in the water the same way we could see a plume of smoke particles in the air. We can't really see where the underwater plume goes, how long it stays in any one place, what it encounters along the way. So this is where modeling and math and oceanography and science come in. Dispersants generally, and the corexit dispersants in particular, have been very heavily researched over the past 25 or more years. The academic and government research communities looked at fate and effects modeling and toxicity assessments and risk assessments and policies of use, all of which employ data to help us understand what happens if we ever do use dispersants. That said, science is an ongoing process. So this project looks at just one small piece of what might be proposed as changes to federal toxicity testing requirements that can also inform changes at the California OSCA licensing level. On the upside, both the federal EPA and California OSPR programs have required some level of acute and chronic toxicity testing for most OSCAs, but some other notes to make here. The EPA program does not currently require toxicity thresholds for any of the OSCA products that it reviews and may subsequently list on their product schedule. OSPR does establish thresholds for effects concentrations of a product when tested against red abalone larvae, but past protocols in the OSPR licensing process were limited to toxicity testing on sorbents. OSPR added the toxicity test on sorbents because the EPA does not require listing of sorbents on the product schedule, so there were no sorbent data available to us from the federal side. So OSPR test protocols give us more information on a sorbent, but those protocols have also been used for testing on other types of OSCAs, including dispersants, and that's not a broad enough use to cover all types of OSCA testing. Generally speaking, and as much as we've tried to make federal and state OSCA reviews align with each other, we still have a lot of work to do. The OSCA data remain inconsistent and incomplete across all categories of oil spill cleanup agents. And dispersants are certainly among these products, and these inconsistencies are keeping OSPR from considering most of the other OSCAs, including dispersants, that are on the current EPA product schedule, but not yet licensed by OSPR for use on California oil spills. We don't want to limit ourselves in the number of products we potentially have available for use, but we also don't want to license products that we just don't yet know enough about. So both the EPA and state are trying to address these issues with their respective updates to OSCA review and approvals, and this COSSEP project is just one step in that direction. So what did the EPA propose as a fix to the federal OSCA review process? Back in 2015, they proposed changes to Subpart J of the National Contingency Plan. Subpart J determines which and how different oil spill cleanup agents are added to the federal product schedule. It includes the test protocols that will be used on the different types of OSCAs to allow their listing on the schedule, and different OSCAs may require different types of tests. So it's crucial to have reliable, quality product information and standardized testing within each category of OSCAs. The changes that EPA proposed making to their process for product testing and subsequent listing on the product schedule would have been much more stringent, comprehensive, and challenging. Each product manufacturer would have had to conduct new tests on their product, and those results reviewed again by EPA before the product could be considered for relisting on the product schedule. The EPA proposal included acute toxicity testing with 2 new reference oils, Alaska North Slope crude oil, and a fuel oil, and their choice was intermediate fuel oil 120. Currently, there are different oils, Prudhoe Bay and Louisiana sweet crude oils, that are required for tests of a dispersant's efficacy. And yet a third oil, number 2 fuel oil, is used for the dispersant toxicity tests. The EPA also suggested using the same 2 oils for both types of testing, the test for product efficacy and the tests for product toxicity. And part of a new suite of toxicity tests would have been those for subchronic developmental effects on purple sea urchin embryos. Testing on ANS would give us toxicity data for the crude oil that's most commonly transported in significant quantities along our shores, and on which dispersants would likely be the most effective. There are actually many types of oils and spill situations where we would not recommend dispersant use, but use on a large spill of ANS might be our most likely consideration. It's also ideal to use the same oils in both types of tests to which they're subject. Efficacy tests evaluate at least the lab scale how effective a given product is expected to be. Currently, a dispersant that is at least 50% effective in lab tests can go on to do toxicity testing. The toxicity tests will be on the product by itself, on the oil by itself, and then on the product mixed with the test oil. Ideally, we at OSPR want to align our OSCA licensing program with what EPA might eventually require so that we can limit how much additional toxicity testing a product manufacturer needs to do. Testing can be costly, and we don't want to recommend tests for which there is no justified need. And that doesn't mean that the state cannot or will not require additional testing when necessary to support our product licensing process and to support the policies of use that will strategically narrow the use of any given OSPR to spills where it will improve the environmental outcome. A few questions for me going into this project was whether ANS and IFO 120 would suit as reference test oils, whether the EPA was proposing the right type of toxicity tests for these products, and whether the purple sea urchin would be a suitable toxicity test species if used as a substitute for red abalone. Urchins are found along many coastal areas, but red abalone are only found along ours. The purple urchin could be a test species on that national EPA stage and provide useful data on a species found in California, then maybe that would be better than requiring separate toxicity tests for a sensitive species that's only found in California. So I got all excited for a while. Policy wonk gets excited and held off on making changes to the OSPR licensing regulations and procedures until I could see what was going to shake out at the EPA level that we could just adopt. But this project turned up additional questions for me to consider going forward. And I'll come back to these toward the end of the presentation. But before we leave this slide, I know it's been a long slog here for this slide. Where does the federal update process stand? So a key date. After November 2016, EPA went quiet on these proposed changes. It wasn't clear how the federal 2-for-1 regulations handicap might apply to the Subpart J updates. Not at all certain that the more stringent testing requirements would have been accepted within our new political climate. So EPA did what was really probably for the best. Rather than push forward for key changes that might not make it through these new challenges, they did what amounted to a strategic withdrawal. And they've not released any new updated material for external review since. In the meantime, we decided in 2017 to use a COSSEP project to explore at least a few aspects of what was in that 2015 draft and see if and how we could move ahead on state licensing updates using, at least in part, some of these project results. So the focus in this COSSEP project was on the dispersant and dispersed oil toxicity testing approach that was offered in that 2015 Subpart J proposed update. You'll be relieved to know that we're only discussing here one tiny outside onion layer of the proposed revisions. So at this point, I'm going to turn things over to Bryn for a while, and he'll discuss the testing methods and resulting data. Bryn, your turn.
All right, thank you, Ellen. My name's Bryn Phillips. As Ellen mentioned, I work for the University of California, Davis at the Marine Pollution Studies Lab, and we are an aquatic toxicology laboratory, and we did all of our aquatic toxicity testing for this project. So basically, Ellen's the policy wonk, and I'm the lab wonk or lab geek. And so what I'll be talking about are the methods for the toxicity testing. I realized recently that our audience today was probably going to be quite broad, and so there's going to be a few slides on what is toxicity testing and some basic descriptions of what it looks like in the laboratory. I'm going to talk about our solution preparation and data analysis, which includes a bunch of statistical acronyms. And then with the results, I'll talk a little bit about quality assurance and the statistical results that describe those acronyms. Next slide, please, Ellen. So we conducted 6 different types of toxicity tests with 2 acute protocols and 4 chronic protocols. Chronic and subchronic are terms that are somewhat interchangeable. The acute protocol, an acute protocol is generally a shorter term protocol that looks at the survival of the organism in the test solution. And a chronic protocol is generally a little bit longer term, especially in the case of something like the mysid shrimp or the top smelt, which is a fish, where we look not only at survival of the organism, but whether or not they develop and grow properly. The embryo larval tests that with purple urchins and red abalone are development tests. And you can see from the photos there that those are what normal looking organisms look like at the end of their exposure period. abnormalities in those organisms would be scored as impairments. All of these tests are listed in this revision, proposed revision of Subpart J. We added the 48-hour test with the red abalone in order to compare our current results with past results from CORSSEP 9500 that have been done historically. Some of those comparisons are a little bit difficult. We'll talk about that more a bit later, just based on differences in methodology. Next slide, please. So I added this slide in the 11th hour to give everybody a little bit of a visual depiction of what a toxicity test looks like and also what the data look like, and to help describe some of the acronyms that I'm going to be talking about. So basically, we create a solution of a dispersant or a dispersant oil mixture, and we dilute that solution into different concentrations. So you have anything from clean water, there on the left represented as a yellow set of beakers that are labeled control, all the way to a high concentration of a dispersant or a dispersant oil mixture that's depicted in red. And you have this range of concentrations, and you want to find out where along that range of concentrations are you going to get effects on your organism. And it's kind of a canary in the coal mine scenario. We use these organisms as sentinel species, as representative surrogate species of what's going on in the environment to organisms that may be exposed to this dispersant or dispersed oil. And somewhere along the line of the concentrations, you're going to get an effect. And if you guys look really closely at those slides, you'll notice that the fish on the right are upside down compared to the fish on the left. Those are the ones that are being affected. And if you look at those data in the form of a graph, that's the bottom part of the slide. You have a control on the left, which is clean seawater, and you're hoping that, you know, all of your organisms will do well in that control. And as you move through those concentrations, you're going to get effects. And somewhere in the middle, you have an LC50, and that's your median lethal concentration. And that's something that we use a statistical analysis for to determine the concentration that affects half of the organisms. If you were looking at these data right here, you would assume your LC50 is somewhat a little bit higher than the LOAC. There are other point estimates like LC50. We have an EC50, which is an effect concentration. We use that for the abalone and the urchins. And we have an IC25, which is an inhibition concentration that we use for the growth endpoints. Next slide, please. So this is a quick summary of our test material sources. It says where all of our organisms came from, where the CORSSEP dispersant came from, and where our Alaska North Slope oil came from. And there's a lot more detail on these things in the report, so I won't belabor them here. But I did want to point out that the Alaska North Slope crude oil, it was pretty difficult for us to find that, and it did delay our project a little bit. We did finally get a hold of some. It's not yet common in the inventories of the research supply houses, and so we had a little bit of a hard time getting that. In terms of the IFO 120, that intermediate fuel oil, we weren't able to get that at all, and so we had to forego testing that as part of this project, which is unfortunate because we could have used those results to compare to the currently used number 2 fuel oil. Next slide, please. Just a little bit on test conditions. All of our tests were conducted at the Marine Pollution Studies Laboratory. And so it's really good to have everything conducted in one place because all of that quality assurance gets shared in one facility and it's not spread out. We're a laboratory that's right on the coast, south of Monterey, and so we have access to clean seawater, pristine seawater, and so all of the seawater that was used in all of these laboratory exposures was from the MPSL site, and it was filtered down to 1 micrometer. It's an ambient salinity seawater. This ambient salinity is about 34 parts per thousand. We have good dissolved oxygen concentrations and good pH ranges in the seawater. This is a seawater that's used as a reference seawater by a number of other laboratories in California. And so it has a pretty good history of participation in projects. Every other thing in our laboratory, because these are toxicity tests that are run in a laboratory, we control every other thing. So things like light, feeding, the number of animals we put in our beakers, the number of beakers we use in our test, the number of replicates are all sort of stipulated by the US EPA protocols that we follow to do these tests. It's kind of like a cookbook, and we follow the recipe. Next slide, please. So I mentioned that we are testing these dispersant solutions and dispersant oil solutions. So there's two different things we're testing, and we have to mix up this dispersant with seawater into a stock solution, and then dilute that stock solution into a range of concentrations that we're going to expose our organisms to. And this is a photograph of the complex machinery that we use to create this dispersant stock solution. We basically start with a liter beaker of seawater with a top mixer. We create a vortex in that beaker, kind of like you're mixing up a smoothie or beating some eggs with your hand mixer in your kitchen. And you add 1 milliliter of dispersant to that beaker of water. And then we can dilute the contents of that beaker of water into different concentrations for our test organisms. And what we're trying to do is we're trying to create a range of concentrations that will bracket the response of the organism. So you don't want to kill all the organisms with all of your concentrations. You'd like to have a couple of concentrations where the organisms do well, and then a few concentrations on the upper end where they're being impacted by the dispersant. So we start a lot of our tests by doing a rangefinder experiment to determine where the optimal range of concentrations is. Oftentimes we know what that range is ahead of time based on previous data, so we can sometimes get it the first time, and sometimes we have to try it a couple of attempts to get that range. But everything we do, all of the tests that we conducted that I'm going to be talking about today, we did a rangefinder test first and then a definitive test second. And so there are two tests for every organism and solution we're working with. I've already described how to do the dispersants in the fourth bullet. So let's move on to the next slide, please. So this slide is going to describe our dispersant oil mixtures. And this is a little bit more complex than creating the dispersants. Imagine, if you will, an oil slick that's on top of a water body. We create our own oil slick in our own portable water body in the laboratory. We use this 20-liter carboy, which is pictured in these photographs. We fill it up with about 19 liters of water. We add an oil slick to the top of that water at about 25 grams of oil per liter of water. So we're adding about 475 grams of oil. That creates a nice slick up on top of the water. We set that entire carboy on top of a stir plate, and this drives a magnetic stir bar that's inside the carboy, and it creates a vortex. Again, much like we were preparing the dispersant, we create a bit of a vortex in that carboy, and then we add the dispersant to that oil slick. And what's interesting is when we first put the oil in on top of the water underneath that oil is completely clear. It looks fine. The water is setting on top of that oil, just like salad dressing oil. And once we add the dispersant, you can see the water beneath that oil slick get cloudier and cloudier as the oil disperses down into that water column. Our dispersant is added to the oil at a ratio of 1 part oil to 10, or 1 part dispersant to 10 parts oil. We tightly put a lid on top to keep volatiles from being released. We mix the dispersant oil water mixture for 18 hours. We allow it to settle for 6 hours. And then we can use that tube there that you see in the bottom photo to drain the oil out from underneath the slick. And that is our CWAF sample. That is the sample that we're working with and diluting and exposing our organisms to. That sample is analyzed for total petroleum hydrocarbons, and then we dilute it for our toxicity tests. Next slide, please. So a few notes on data analysis. I mentioned some of the statistics back in that toxicity slide, our point estimates like the LC50 and EC50, and also the NOx and LOX. We calculate these in a couple of different ways for our two different solutions that we're testing. The statistical calculations for the dispersant-only tests use the nominal concentrations of the dispersants we've added. And so those are part per million concentrations. There isn't a good way to easily measure the dispersant in the water. So you have to use a nominal concentration, which is the concentration that you've intended it to be in parts per million. With the dispersant oil mixtures, we actually use the total petroleum hydrocarbon concentrations of our test solutions to calculate those statistics. So you'll see in the results slide that they're slightly different units that we use to present the results. Next slide, please. A few notes on quality assurance. Quality assurance is the basis of any laboratory data, and you definitely want to review all of the QA data that has come out of your tests to make sure that your data are considered acceptable for interpretation. Any good monitoring program or science program will do this with their data. And so what we do with the toxicity data is we look through and we make sure all of our organisms had acceptable control survival, acceptable control development, and acceptable responses in reference materials to make sure we know that those organisms performed exactly like they were supposed to, that the only reason they would have behaved differently, passed away, developed improperly, any of those things would have been only due to a toxicant that we would have introduced, in this case, the dispersant or the dispersant oil mixture. So with our survival and development and growth endpoints, everything was good for our toxicity tests except for one of our top smelt tests, which had a low biomass in the control. We considered that test to be acceptable for using for data interpretation because it still had a really good dose response, and that dose response was in the rangefinder test and not the definitive test. So we went ahead and didn't repeat that test. All of our water quality parameters were within acceptable limits. The EPA protocols have recommended ranges for the water quality parameters. Some of our salinity values were outside of those recommended ranges in the MICID test and the top smelt tests. And because those organisms can be tolerant of a wide range of salinities, we still considered the data acceptable for use. We also had a few issues with our reference material tests. Some of them didn't perform as expected, but all of the controls were acceptable. And again, we considered the data acceptable for interpretation. On the analytical laboratory side, the work that was done at the Water Pollution Control Laboratory in Rancho Cordova, all of their analytical quality control criteria were met. And everything looked good, so we were able to interpret those total petroleum hydrocarbon numbers with confidence. Next slide, please. Okay, now we're going to get into the thick of the data. So I'm going to orient everybody to this slide so you can understand what's going on here. There's a lot of numbers to look at, but I'll take the time to point everything out carefully. The first column lists our species and tests, and the acute tests are at the top, so those only have a survival endpoint, and Americomyces bahia is our MICID, Atheranopsis affinis is our top smelt, and we use those same two organisms in a chronic endpoint as well. So we have a 48-hour test for MICIDs and also a 7-day test. We have a 96-hour test for top smelt and also a 7-day test. You'll notice that with the 7-day tests, there are two endpoints. If you move over to the third column, the endpoint column, you'll see that there's a survival endpoint and a growth endpoint, or in the case of top smelt, survival and biomass, which is very similar to growth, just calculated a little bit differently. For the embryo larval tests, we look at development instead of survival. All of the duplicate tests are presented, rangefinder and definitive, and then the columns to the right, the NOEC, LOEC, point estimates, results, and averages are all of the results of the statistical analysis. So I want everybody to kind of focus on the point estimate results, which are the second to last and last columns there. So basically the very final column, the mean result, is just an average of the two numbers in the column to its left. So the thing that we're most interested in, what I'm usually interested in as a toxicologist, is what was the most sensitive organism, you know? And so I look for the lowest number in that final column. And you'll notice that the lowest number is under Haliotis rufescens. That's our abalone test. And it had an EC50, an average EC50 of 6.28 parts per million. So the lower the number, the less sensitive the test, or the more sensitive the test. And the higher the number, the less sensitive the test. The highest numbers depicted in this table are for the top smell tests, a Theranops affinis. It had the two endpoints, the survival endpoints of both the long and short-term test, the acute and the chronic test, were in the 60s. So the acute test had a 60.2 average, and the chronic test had a 67.2 average. So those are the least sensitive of all of the organisms we tested. With the MISA test, usually when you have a shorter term test, it's going to be a little bit less sensitive than a longer term test. Imagine, if you will, the longer you're exposed to something, the more likely it is you're going to be affected by what you're being exposed to. And with a 48-hour exposure, it's a pretty short exposure, and that's the acute exposure for the MISA test. You'll notice that the mean response was 38.8 parts per million, whereas the chronic side of that was 27.1. So this is completely expected. We expect that longer exposure to have a lower number. It was interesting to note, though, that the top smell tests were sort of the opposite of that. The short-term test had a lower number than the long-term test, 60.2 versus 67.2. Those are both sort of within the realm of error. If you'll notice, with the results, you'll notice that the numbers they're averaged from have a somewhat overlapping range. So it's not completely unexpected to see those types of data when you've only done two tests. The urchin test, which we're hoping to have a similar sensitivity to the abalone test, was actually quite a bit less sensitive than the abalone test. The urchin test is down there at the bottom, S. purpuratus, and it had a mean response of 29.7 versus the abalone test of 6.28. Can I have the next slide, please? So I'm going to talk a little bit about the dispersant oil results now, and I have to make a point of informing everybody about our total petroleum hydrocarbon correction factor. When the laboratory first measured our dispersant oil mixture, they found quite a bit of interference in the sample. It was very difficult for them to tease apart the hydrocarbons that were in that sample from the oil versus some sort of interference that was coming in from the dispersant itself. And so what we did is we actually created a dispersant-only sample for the laboratory to measure so that they could create a correction factor for the dispersant oil mixture. So basically it's some number that you would subtract from your final TPH concentration from your CWAP sample to get your interpretable number that you can use in your statistical calculations. So it was a good thing we kind of figured this out early on, and we were able to create that correction factor, and we were able to get final total petroleum hydrocarbon concentrations of 638 and 483 for the two CWAF tests that we're using to interpret these data. Those numbers actually, to me at first, I thought they were kind of far apart. You know, I would have expected two samples that we'd created in exactly the same manner to be somewhat closer in total petroleum hydrocarbon values. But it's definitely within the realm of variability for these types of samples. Next slide, please. So this next slide is the results of our dispersed oil toxicity tests. So these are our CWAF tests, and the slide is structured in the same manner as the data from the dispersant only tests, except what you're seeing in this slide are basically just the top rows of the previous slide. We only did the acute toxicity tests for our dispersant oil mixtures. So we have our 48-hour MISA test and our 96-hour top smelt test. Again, we have a rangefinder and definitive. These are survival endpoints only, so I don't have an endpoint column on this slide. And the first thing I want to point out in this slide was our rangefinder test for MISIDs was actually quite a bit off. And in our first exposure, we weren't really able to calculate a NOAC. That's why it says less than 19.9 under the NOAC column there. And our result for the LC50 of 7.77 was extrapolated using a statistical program. So it's not the most accurate LC50, but it is usable data. We were able to hone in on the concentration range much better for the definitive test and get good statistics for NOAC and LOAC and a more accurate point estimate of 12.9 milligrams total petroleum hydrocarbon per liter. And that's an average of those two numbers of 10.3 TPH per liter. Our top smelt test was quite a bit less sensitive than the MISA test, as we would have expected from our dispersant only exposures. It's about 7 times less sensitive. 70.3 was the average LC50 in milligrams TPH per liter versus 10.3 for top smelt. Next slide, please. One of the requirements of reporting under subpart J is to create a summary table. And so even though I've gone over these slides and shown everybody what the results were in pretty good detail, I wanted to just sort of show all those numbers together to give a bit of an easier indication of where the organism hierarchy was. The top part of this table includes all of the dispersant only tests. And if you look at the final column under mean concentration, you'll note that those results are in parts per million of dispersant. And under the CWAF tests, or dispersant oil mixture, those results are under milligrams per liter TPH. And again, just to reiterate, the abalone test was the most sensitive in the dispersants at 6.28 parts per million dispersant. And our acute MISID, which was the second most sensitive in the dispersant test, was the most sensitive in the CWAF tests, 10.3 versus top smelt of 70.3 milligrams TPH per liter. The data comparisons with our historic data, which Ellen's going to discuss in a little bit, are a little bit confusing because we are comparing a static protocol where the organisms were exposed to the same concentration of dispersant or dispersant oil mixture for the entire duration of the exposure versus other methods like an exposure recovery method or a spike method where organisms are sort of exposed to the concentration of dispersant for the initial part of the protocol, but they're allowed to recover over time in clean water as the exposure progresses. So some of these results are a little bit different from what we might see in historical data. Ellen, I'm going to turn it over back to you. Thanks for clicking the slides.
Thanks. So what I'm going to show here are some toxicity test results for Corexit 9500 as it currently appears on the EPA product schedule. This product schedule is updated every few months or so. So this slide, it shows toxicity results for dispersant by itself, the number 2 fuel oil by itself, and then the dispersant when it's mixed with the number 2 fuel oil. And as Bryn mentioned, this is usually on a 1 to 10 ratio, 1 part dispersant to 10 parts of oil. So remembering that higher numbers mean a lower toxicity, we see that the Corexit by itself was less toxic than the number 2 fuel oil by itself, but mixing the two together was where you kind of get the toxic combo. Some other differences to note with this table, this is a different test oil than was used in this COSSEP project, and there was also a different test species. This used inland silverside as the fish rather than top smelt. And then just some other notes to make, trying to understand tables like this and what it really means. When you mix a dispersant with oil, it results in greater toxicity, but it's from the oil components because they're now more available to the upper water column species. Now that dispersed oil, the little bitty bite-sized pieces can now also cross over membranes and go past fish gills and things like that. But here's what I really want to emphasize, and you get quotes here that you get to apply to what I'm about to say. It's not what they are, it's what they do. So an effective dispersant, and by that, something that effectively takes oil from the surface and delivers it into the water column, even if it's absolutely non-toxic in and of itself, it still delivers toxic oil particles into the upper water column where a host of different species and life stages are now potentially exposed. So the balance we try to hit in our response choices, our oil spill response choices, knowing that every choice is going to have its pros and cons, is to determine in advance, as much as we can, what will be exposed and what life stage is the most sensitive, how great that exposure will be in terms of concentration and how long it's going to last, its duration, how the organism or life stage handles that exposure. Different critters and life stages handle exposure differently. And if the exposure does result in death, what does that mean to the local population and its recovery? I'm going to put up these two toxicity information boxes up here. They come from 1995-96. And from when Corexit was initially put on the EPA product schedule and its initial license with us, both dated from 1995 and 96. So here, different species, again, Fundulus heteroclitus is the fish mummichog, and Artemia salina is a brine shrimp, not too dissimilar from the mysid. But I think it's interesting that in the middle entry in the left-hand box, the mummichog is showing a really high tolerance for oil. They're really hardy, which is why OSPR pushed, even back in the mid-90s, for going beyond the usual suite of test species and required for our OSCAR licensing program that toxicity testing also be done on red abalone larvae. Red abalone are a California endemic species, and while the adults living on the ocean bottom might not be exposed to large amounts of dispersed oil, their larvae that are floating at the water surface, they will, and they're exquisitely sensitive. We required toxicity tests to determine the levels at which the abalone larvae would show problems with their early shell development if they were exposed to dispersant and dispersed oil. However, our OSPR red abalone toxicity test protocols are currently being used for testing against all types of OSCAs, including dispersants, when they were written to direct the testing of sorbent products. And that testing required creating a leachate of the sorbent product and testing that in a 1 to 100 dilution with seawater. So different dilution ratios means the test protocol really cannot be directly extrapolated for use with other OSCAs. So my backyard weed patch looks a little bit better. Did we get everything from this study that we needed to kind of make our our OSCA licensing process a little more orderly? And the answer is, yeah, we made some progress, but we also turned up more questions. So what's that mean for our recommendations going forward? I'm gonna have you revisit that earlier busy slide, but since we already looked at that box to the left, that kind of peachy colored one, I'm going to focus instead now on the green box to the right. So we'll answer those first two questions and reveal the three additional questions. So let's look behind door number one. So COSSEP stands for, this is why acronyms are handy. It stands for Chemical Response to Oil Spills Environmental Research Forum. Reaching back 30 years ago and into the late 1980s, much of the dispersant and dispersed oil toxicity data in the literature was based on these typical static exposures of 48 to 96 hours in duration. The static exposure tests on dispersants were criticized as potentially overestimating the toxicity of oil and dispersed oil in an actual spill scenario. So in response, COSSEP, the COSSEP working group, which included academic, industry, and government scientists, developed toxicity test protocols involving spiked exposures of shorter durations, because that appeared to be a better model for how exposures might actually play out when dispersants are applied to a surface oil slick in the open ocean setting. So the COSSEP protocol standardized the methods to allow for greater comparability of toxicity data, as well as relative toxicity of different dispersants in oil and the relative sensitivity of test organisms. And after that, there were several additional studies on dispersants funded by OSPR that were also done down at the Marine Pollution Studies Lab, where Bryn is now. That research involved different types of oils than ANS or IFO 120, and looked at two other COREXIT formulations in addition to COREXIT 9500. But it did include toxicity tests on the red abalone, but not the urchin. So I want to make a special point here. It kind of underlies this whole presentation and the need for a COSSEP project and changes going forward. It's really critically important that we keep the apples to apples maxim in mind here. We can't directly compare toxicity results from one testing protocol regime, such as acute tests using a static exposure over several days, to a different protocol regime, such as the COSSEP model with a spiked exposure that occurs over a shorter duration. Trying to compare data from different studies that use different oils and or different species and or different protocols. It's been a huge problem in the past. COSSEP tried to address this, but we need to further incorporate the COSSEP lessons learned as we go forward. And back up to one of those original questions, we wanted to investigate whether purple urchin could be a suitable alternate toxicity test species for purposes of state licensing. And the data from this project indicate that red abalone is still the more sensitive species. So there's a need to keep that in mind. This COSSEP project used top smelt as the test fish species rather than the inland silverside fish recommended under the current subpart J. The data from this project showed, as Bryn mentioned, top smelt with an LC50 of around 60 parts per million in the acute test. While the data from the EPA product schedule listing for COREXIT shows an LC50 of about 25 parts per million in the acute test on the inland silverside fish. So it may be that the inland silverside is the more sensitive species, and maybe we'll suggest that EPA just kind of keep that as their fish test species of choice. This COSSEP project should probably also forward the question about whether a correction factor should be used for TPH measurements. As Bryn alluded to, some dispersants, including COREXIT 9500, have hydrocarbons in their recipe. So in this project, the hydrocarbons were supplied by the dispersant alone, were subtracted from the TPH totals to give a TPH for the dispersed oil by alone. So Should EPA consider the same in their proposed updates, at least for the dispersant formulas that include hydrocarbons? And if they don't, should we? So that's some of the recommendations. Now, let's continue on this path and look a little more at the recommendations. And I got to say, Piglet, I think Piglet is one of my early heroes. I hope you'll continue to mentor me as I continue to work my weed patch and push forward on some of these recommendations. So for the EPA, we'd recommend using the same oils for both the efficacy and toxicity sets of tests. If using new reference oils such as the Alaska North Slope and IFO 120 suggested by the EPA in 2015, then make sure that there are reliable quantities of well-characterized oils stocked by the research supply houses and available to all the people that need to test against those oils. And if it can't be assured that IFO 120 is a fuel oil that can be mixed, kind of gets mixed at the pump in a way. If it can't be mixed to provide a standardized and consistent set of properties, then maybe we just continue to use number 2 fuel oil as the reference fuel oil for testing. And we'd recommend that EPA consider requiring COSSEP protocols for the dispersant toxicity testing. So this is the much more relevant toxicity test protocol that we feel should be routinely used going forward. That said, this greater utility might only apply to dispersant toxicity studies, while the static exposure and longer duration tests, those 48 to 96-hour tests, may still be the better test for other types of oil spill cleanup agents that would be applied to the environment in different ways than dispersants are. From the OSPR side, we need to broaden our red abalone larval test requirement to apply to other OSCA product toxicity testing as well, not just sorbents. And even if EPA doesn't require that some version of the COSSEP protocols be used for dispersant and dispersed oil toxicity testing going forward, OSPR probably will. And I'd recommend holding on to the red abalone larval testing requirement for our own licensing program, at least for now, as they appear to be the more sensitive invertebrate test species when compared to the urchin. And again, we want to look at what's most sensitive so we can be conservative in our decisions about how to use cleanup agents. There may be advantages to another round of testing using the COSSEP protocols, focusing again on urchin and abalone larvae and comparing those toxicity data with each other. And while the subpart J protocols suggest a continuation of testing at that 1:10 ratio of dispersant to oil, which I'm not averse to because it does give us an apples to apples benchmark, it might be valuable for California to also run the COSSEP protocols on urchin and abalone at the 1:20 ratio. The 1:20 ratio is the initial dilution recommended in applications of Corexit 9500. It's also a condition of the Section 7 concurrences that were conducted under the Endangered Species Act, that dispersant be applied in a California spill response, at least initially, at no greater than a 1:20 ratio. Okay, that's it. That concludes our presentation of this COSSEP project. And if there's still time, we'll let Brian channel some questions toward us. If you know which of us you want to direct your questions to, let Brian know. And any questions not answered today or that you think of later, please feel free to email Bryn or me. And from both Bryn and me, thanks for your attention. Many thanks from me to Bryn and to Martise for all their work on this project. Toward the end, both of them had to run some additional analyses in a time of COVID, and extra points to Bryn. He had to deal with big things like repairing the Granite Canyon seawater system before he could even get started on this. So thanks, Bryn and Martise. Thanks all of you, and thanks all of you listening today. Brian, now it's your turn.
All right, thank you, Ellen and Brand.